Okay, ketone bodies are a group of molecules that can be used for energy in most cells, but particularly in heart, muscle, in the adrenal cortex, or during starvation. So they result from an overproduction of acetyl-CoA from beta oxidation. The brain can adapt to using the ketone bodies after starvation. So we're getting a lot of acetyl-CoA from beta oxidation, and what can we do with that acetyl-CoA? So if you have a high intake in lipids and low in carbohydrates, so what diet is that? High lipids, low in carbohydrates. Is it the Atkins?
Yeah, I think it's high protein.
It's mostly
high protein?
Ketone.
Yeah, high protein.
But don't you end up eating a lot of fat too, though, if you do?
Eating a lot of steak and stuff, you're eating a lot of fat.
Yeah, if you're eating a lot of steak, you're going to be eating a lot of fat. So high intake in lipids and low in carbohydrates, you're going to have ketone bodies result. So these are these three molecules. You need to memorize these three, what they result from. If diabetes is not controlled well, during starvation, you're going to be going to the fat in your body for the energy. So actually, you can actually smell the acetone in people's breath during ketosis. It's going to overwhelm the buffer in your blood and disrupt your sodium and potassium in your blood. And what you're going to happen is you're going to dehydrate and can be fatal. So that's one of the things that you healthcare workers are going to look for in uncontrolled diabetes is the and results from the overproduction of acetyl-CoA.
It's only 40% of the population can smell it.
Only 40% of the population can smell that? Really?
So only some people can smell it, others just
So only 40% of the population can smell the acetone.
smell the ketones, yeah.
Smell the ketones. So that should not be a test. That
It's not a good test.
Not a good test.
You can do it.
So maybe people that live with people with diabetes should see if they can smell it or not, right? Wow.
We did a thing in our neuroscience class once where we did a bunch of different ketone bodies and like different scents and stuff, and like half the class could smell certain ones and the other half the class couldn't smell other ones.
Okay, so that supports.
Yeah.
Does everybody who has diabetes have the smell or just
If their
uncontrolled?
sugar's above,
Yeah, this is from uncontrolled diabetes.
You could smell it.
You can smell it. If you can smell it, you can smell. You can
I
listen.
worked in a lot of patients with diabetes, and if they would come in before the study and they weren't ready, you can smell it on their breath.
Wow. So I guess if you're a healthcare worker, you should know whether you can smell it or not. And yes, Patrick.
I'm not sure what the process is, but for the low carb diets, they can use it to control epilepsy in some children. That's where a
They
lot
can
of-
what? I can't hear you.
They use it to control epilepsy in some children. So that's where all the research on lower carb dieting has come from.
Oh.
For the most part.
Yeah, I don't know where that would
Yeah,
come
I'm
in
not
here.
sure the process is.
Wow. So let's take a look at how does an overproduction of acetyl-CoA result in these three molecules, guys. You don't need to know the structure, but these three molecules are our ketone bodies. So here what we're doing is we're condensing two acetyl-CoAs. We're condensing two acetyl-CoAs to get acetyl-CoA. You don't need to know that. But what we can do is we can then cleave this coenzyme A off, and you get acetoacetate. That is one of our ketone bodies. You can reduce this carbonyl to an alcohol to get beta-hydroxybutyrate. And then if acetoacetate is decarboxylated right here, then you get acetone. So I'm just showing you how they are related to each other. What I want you to know is what do they result from? Overproduction of acetyl-CoA, and what their names are. So you can see acetoacetate and beta-hydroxybutyrate are related by this reduction, and acetoacetate is related to acetone by the decarboxylation. And for fun, I just put the structures up there, the ball and stick. I thought it was pretty. All right, we already covered this, but we're going to just mention it again. Atherosclerosis, again, the lipids are transported by lipoproteins. Remember, the lipoproteins are a ball of phospholipids. When inside you have your triglycerides, you have your cholesterol, and then you have an apoprotein. And whether you're LDL or HDL depends on how much protein you have. Remember, protein, I'm starting to lift again. All right, so, and I thought it was wise to run 3 miles last night and 3 miles this morning again. Not very wise, because I'm sore standing up here. So that was not very wise. But I'm hoping that I actually gain weight because I will have more muscle. So remember that. High-density lipoprotein is going to have more of the protein. That's why it's good. It has less of the lipids. And then low-density lipoproteins are going to have more fats than the protein. Again, this is our pretty picture. This is our apoprotein. I don't know why it has little mountains on it, but this is our apoprotein. Remember, an apoprotein is a protein without the prosthetic group, so I guess this whole thing. You have your phospholipids, and you have cholesterol and other fats inside there. So this is just our carrier. Remember, cholesterol is not water soluble, so how are you going to get that around your blood? You got to find some way. So what you do is you cover it in a ball of phospholipids. Very intelligent. And again, this is the same slide that we showed before. Our endocytosis, this is how we're going to get cholesterol into our cell. If we have overproduction, we're going to then get that receptor inhibited. All right, oxidative stress. I mentioned that it's not in your textbook, but I do think it's important just to briefly go over it because we live in such a reactive environment. In fact, when you start studying this, you think it's kind of amazing that we do survive in such a reactive environment. And what we have is 2 ways to protect against reactive oxygen species. So oxygen is super reactive. And so the way when oxygen can react with something, it can produce these molecules that are particularly tough on our cells. And we're going to take a look at how they are tough. So these little dots here, that means they're radicals. And we're going to see what the radicals can do. Hydrogen peroxide is partially reduced oxygen. When we have fully reduced oxygen, it's water. So this is partially reduced oxygen. And we can get the radical from that. So ROS stands for reactive oxygen species. We have antioxidant defense systems. And they're either small molecules or enzyme systems. And we're going to cover both of those. But there's 100 diseases linked to our failed systems. So, and so we can get a lot of diseases from living in our oxygen environment if we're not protected. So we can also get them from, this is just super weird, electrons leaking out of the electron transport chain. That's just weird, okay? Exposure to UV light and radiation. All right, radiation, so if you're near Chernobyl, this might happen. I just listened to a report about the animals in Chernobyl are doing great. So they're, it's like this nice environment for them because there's no people. So they're showing that it's better for animals the wildlife to live in nuclear radiation, high nuclear radiation, than to live around people. So that's horrible. Isn't that terrible? UV light, I'm hoping to get some of that today. side. Anyway. All right, so do you guys remember when you're doing your work in Gen Chem 1 that you learned that oxygen wasn't really a double bond. It was actually, this was the electron dot structure of oxygen because it was paramagnetic, and that you needed the molecular orbital theory to describe oxygen. This is what oxygen really is. So oxygen can really easily pick up an electron. Oxygen is not O2 double bond. It's not. Oxygen is actually paramagnetic. It's magnetic. If you pour liquid oxygen between poles of a magnet, it will stay right there. It's attracted to the magnet. And it's because the electrons are here. Do you remember the levels of molecular orbital diagram? Remember those a little bit? No, that's not molecular orbital. That's atomic orbitals. So that's different. It's a little bit different. The sigma and the pi, those are the molecular orbital. And we sometimes professors don't cover it. Yeah, 100 years ago. Okay, so it can easily. The point is that oxygen can easily pick up an electron, making this superoxide radical. All right. So I just want to point out, if you have that superoxide radical, we can get this singlet oxygen by reacting with protons. We can also get this hydroxyl radical. We're going to focus on this one, this hydroxyl radical. What can happen with this hydroxyl radical? It's not so nice. What's really neat is that these reactive oxygen species are actually produced, even though they're terrible. They're produced by macrophages and neutrophils, and you can think of them as bullets. Reactive oxygen species are bullets, and so these macrophages, what are they? Their job is to protect your cells against foreign or stuff that we don't want. So you can think of these reactive oxygen species as bullets. Sometimes you want to use bullets to take care of bad things. I'm not promoting gun violence. So weird. So here you're using the bullets to get rid of things that you want to get rid of. So the macrophages will actually put out reactive oxygen species to kill foreign microorganisms or to get rid of damaged cells. So that will happen. But often we don't want it. So we have, this is what happens. So here we have a fatty acid. Say it's a part of your membrane, right? We have this. What happens is this hydroxyl radical will give an electron to the carbon that's next to a double bond. And so then we have our fatty acid chain with this radical. It reacts with another oxygen and makes this peroxide radical. And what happens is then another lipid, this L stands for lipid, the radical ends up going onto this new lipid. This one has the radical now, and it gives a hydrogen to that. You have metals that come along, and then that turns it back to a radical. And what happens is then it gets cleaved. So essentially, what I want you to know is that by putting a hydroxyl radical, which is one of our reactive oxygen species, it will pass on that radical, that lone electron, and end up cleaving your fatty acid, chopping it like a bullet. But this is a chain reaction. It's a chain reaction because look what's happening. We have another radical here. This one that I showed down in these 5 steps, that's where it got chopped, the original one. But then we've given it to another lipid. And that one's going to get chopped, right? And then that's going to give it to another one. And that's going to get chopped. This